For the first time in 22 years, we have new information on the Roosmalen's dwarf porcupine. In today's video, we are going to break down the publication that was recently published in Zookeys, talk about the phylogenetic placement of this species, and the conservation challenges it is facing. Timestamps and sources are found in the description, and if you want to show support for all the content that I'm producing here on YouTube and Instagram, consider supporting me on Patreon. With that, let's get into the paper. So this article from Mongabay crossed my newsfeed this morning, and it is titled Scientists Unearth Secrets of the World's Smallest Porcupine Species, which is this one right here, Roosmalen's dwarf porcupine. Although described by Western science in 1996, there really hasn't been much information on this species. This species is found in the Madeira biogeographical province in Brazil, where it might be an endemic species, meaning it's only found there and nowhere else on planet Earth. This is an arboreal, secretive species found in the trees. You can see here, this picture, they do possess a very long tail, which they use to grab onto the branches. In fact, there is a photo, this photo right here, where you can see this tail wrapped around the branch. Many arboreal porcupines exhibit this trait, and this is no exception. However, of the 16 species in this genus, Quindou, I cannot pronounce things, 6 of them are so understudied that their conservation status is still unclear. This would mean that within IUCN, the organizing body that says if a species is endangered or not, they would be listed as data deficient or not evaluated. And to compound matters even worse, we are unsure how these species are related to one another. Many are lacking molecular evidence, and this was also the case for Roosmalen's dwarf porcupine. Unfortunately, a bulldozer at a mining site within the species range did kill one of the individuals. It was a very small individual, measured 23 centimeters, about 9 inches long. The tail was about the same length as the body. Fortunately, they were able to get this species and actually extract molecular information so that we may better understand how these species are related. All of the methods and results are detailed in this paper published in Zookeys, Phylogenetic Relationships, Distribution, and Conservation of Roosmalen's dwarf porcupine. Now, this paper does show some of the past specimens collected. Unfortunately, these past specimens, they were not able to collect DNA from, but we see that we do have skulls, mandibles, as well as preserved specimens, both as stuffed skins and as a fluid-preserved specimen. To determine how the Roosmalen's dwarf porcupine is related to other species within the genus, they used mitochondrial DNA as well as morphological characteristics to place it on a phylogenetic tree. Let's break down what that means. This is going to be a very brief overview of phylogenetic methods. I'm not going to spend too much time, but I think it's important for us to understand how they are built. So whenever I say a phylogeny is simply a tree, a bifurcating tree that tells us how species are related to one another. They can be drawn like this. I have a whole video on how to read a phylogenetic tree. Check the card up there. But let's say we have species A, we have species B, and we have species C. Here, we can see from this tree that species A and species B are most closely related to one another because they share this node here. And then species C is most closely related to the group containing species A and species B. But how do we actually build these phylogenies? How do we actually get this information into this tree? And the way we build these phylogenetic relationships is by looking at the similarities and differences between the species. So say we collected genetic information for all of these taxa. So species A, species B, and species C all had genetic information. Let's say species A had some of the most basic information in the world and was all A's. Just note that this is very unlikely in practice, but we are just doing an explanation here. So species B is, it's most closely related to species A. It's very similar. So it may be mostly the same, but have a few key differences. Maybe instead of a, we have a G, right? We have a G there. So only one difference is separating these two taxa. Again, very, very simple explanation. Most cases you're using thousands of these nucleotide bases here instead of 6. But species C, being much, much, much different than species A and species B, may look wildly different. It could be GG, AA, it's not entirely different, but it in essence has more differences than between species A and species B. This is, again, very, very simplified, but it underpins the whole process of phylogenetics, where we are looking at how similar and dissimilar different information is from one another. So, what did the authors utilize? So the authors utilized mitochondrial DNA. So again, genetic information like we have here, AAA, GG, CTT, whatever it is, they used that information to build this phylogeny, as well as morphological characters. So in the absence of good genetic data, mitochondrial DNA is a start, but that's only one gene. They supplemented it by using morphology. I'm always saying that morphology does not mean it is perfect, but that means that is the data that we have, and that is the data that they are going to use. So here are different quill and bristle quills of different species in this genus. And we can see here that A, Longicaudata, is quite long. It has yellow, it has black, it has white. But we see another species here with B, C, Baturitensis. Baturitensis, I said it right, has a cream-colored base with a brown, largely brown quill with a white tip. So they can look at the color, they can look at the size, they can look at the morphology of these quills, and using that same methodology of differences between them, they are able to create a matrix of differences that they can use to build a phylogeny. They used more than just quill. They used the skull shape, they used the size of the animal, I believe they also used the tail length, but in essence, they are utilizing as much information as they have available in order to build a phylogeny. And this is really important for helping us understand the evolutionary history of this species, as well as developing more studies so that we can protect it. I am unable to go in depth on phylogenetic methods in this video because that could be a whole video series on itself, but I highly recommend reading the paper to understand the phylogenetics of this species and what they did. But from now, we're going to talk about the conservation of this taxa. The authors ended up placing this species within the subgenus Caaparamus, which again, there's really not many other species, maybe only 5 or 6, and they are still not fully well described. As I mentioned, many of the species in this genus do not have great information. And that's about all I'm going to say for the phylogenetic aspect. You should read the paper to understand more of what they're doing. Phylogenetics could take up a whole video series by themselves, but now I really want to dive into the section distribution and conservation of C. roosmalenorum. So this figure shows the distribution of C. roosmalenorum. It's actually much wider than they originally anticipated, which is a good thing for the conservation of this species. What they did was they utilized extent of occurrence and area of occupancy. So they did build out both of those. Let's explain those very briefly. So the authors calculated extent of occurrence and area of occupancy. So to explain these briefly, extent of occurrence is the total area that encompasses all known areas that this species has been found and is predicted to be found. So using lots of information, you can draw an extent of occurrence that maybe looks like this, right? So this is the theoretical range, the theoretical area that an organism can exist in, a species can exist in. The area of occupancy would then be the realized area. It is the area within the extent of occurrence where the species actually exists. So in this case, it may be a portion here. You can absolutely have disjunct ranges, meaning they are not connected. You can have portions going up to the boundaries. But yeah, so this would be area of occupancy. And if you think about it, like let's say there's this total extent of occurrence, bordered by the pink here, and perhaps right here is a soybean plantation, right? That is clear cut out the area, clear cut out the forest, And now this is no longer suitable for that organism in question, hence why the area of occupancy is so different. And there could be many other reasons why the area of occupancy is not matching the extent of occurrence. Say there's mountains or rivers or another co-occurring species that causes it to, they have some type of competitive exclusion going on, meaning that one is going to push out the other species. So there are natural as well as unnatural ways that area of occupancy can differ from extent of occurrence. Luckily, from the extent of occurrence and area of occupancy, they actually say that it's likely underestimated due to sampling gaps. They just don't have all the information necessary in order to say, hey, the species is found everywhere. However, they've also lost around 10% of the forest cover replaced by pastures since 1987, and they have lost approximately 15,000 kilometers, over 15,000 square kilometers of forest cover between 2008 and 2022. Given this is an arboreal species, meaning it is in the trees, they do need this forest cover. It is almost certain that pasture land is not suitable habitat for this species, so any areas that are clear-cut for pastures no longer is, well, suitable for the species. And all of this information, although it is preliminary, although the authors themselves very readily admit that it is underestimated or not sufficient, the phylogenetic information, these updated geographic information, is critical to the conservation of this species. And the authors do say that they need to collect more information, listing out a variety of approaches, such as telemetry, arboreal camera traps, literally traps set up in the trees, visual census, meaning just going out looking for them, but also using methods such as thermal drones, which are really efficient and can survey for arboreal species. It's been used in large primates as well as other arboreal mammals, but they also call on citizen science data. Mainly animals photographed by the community may provide new records. This is always something I'm going to support and promote heavily. We run international bioblitzes because of how important it is for studies like this. You going out into nature, taking a photo of an organism with your phone can have instant use for conservation. This is something I've always been very, very supportive of. Citizen science data has been extraordinarily useful for my own research. I've actually published research that uses this information before, and I think it's something that you can directly get involved with. We do run international bioblitzes, and it is for this particular purpose, to encourage people to go out and to find organisms to help conservation. But that is all I had to say on the Roosmalen's porcupine in this amazing study. Again, links are in the description. You can read this paper yourself, understand it further. And if you liked this video, if you want to support what I am doing here on Instagram, YouTube, and other social medias, consider liking, sharing, subscribing, doing all the things that YouTubers tell you to do with this video, and also supporting me on Patreon. Thank you. Have a wonderful day.